want to welcome everyone today to our monthly Earth webinar series. And we are, well, every month is an exciting month, but this month is also very exciting because we're going to have a presentation on some work by Dr. Dimitri Abrahamsson, who is an environmental chemist who's going to talk about his really novel work collaborating with us and our other partners on using non-targeted analysis to uncover the exposome and its relationships to adverse health outcomes. So we're super excited because Dimitri came to us from a postdoc at US EPA in the National Exposure Research Laboratory. And before that, he got his PhD in environmental chemistry at Stockholm University. And he's very expert in chemistry. He studied partitioning and persistence of volatile methylocytoloxanes in the environment. So if we have time at the end, you might ask him about siloxanes because it's a personal pet project of him, and he's done some work with us at PRI on this. And so Dimitri's been a postdoc with us for maybe a year or two, and he recently was awarded a National Institute for Environmental Health Sciences K99 postdoctoral scholarship. And so he's going to talk about some of his work in this area, which is very relevant to the work we're doing in the Earth Center, which is trying to understand and address multiple environmental chemical exposures, particularly those that occur during pregnancy and early development. So I'm going to turn it over to you, to Dimitri, and thank you so much for speaking with us today. And please put your questions in the chat as they come up, and we'll have some time at the end to answer those questions or talk about siloxanes or whatever else comes up on people's mind.
All right, thank you, Tracy, for this awesome introduction. So I wanted to start maybe with that idea of the exposome, because it's probably a buzzword you've heard before. And I thought maybe it would be good to explain what that concept means and what it encompasses. So scientists came up with that idea of the exposome as a way to describe the totality of our exposures. And that became prevalent when we started realizing that genes don't really explain everything, that there are other factors that play an important role in the development of human disease. Recently, we've had some important advancements also in high-resolution mass spectrometry and analytical chemistry in general that have enabled us to screen biological samples for a very broad spectrum of chemicals that would previously remain undetected with traditional targeted analytical techniques. And now with these new tools, we are able to basically characterize the chemical parts of the exposome, and meaning essentially what are the chemicals that we are exposed to, how many of these chemicals are there, and are they interfering, and in what way are they interfering with our metabolism and with our bodies. And as a chemist, I'm very interested in the chemical aspects of non-target analysis, and I'm also particularly interested in the molecular mechanisms behind the development of human disease. Yep, there we go. Next slide. So one of the questions that we are struggling with is how many chemicals are we actually being exposed to? And you'd think that this is a trivial question, but it's actually very complicated. If we have a look at the Toxic Substances Control Act of the Chemical Substance Inventory, we see that there are about 80,000 chemicals that have been registered under that inventory, out of which 40,000 are actively used in the United States. It's important to mention here that the inventory does not include pesticides, food additives, drugs, chemicals used in cosmetics, chemical used in tobacco products, nuclear materials, and munitions. So it leaves out some very important groups of chemicals. Then when we look at, for example, the database that EPA has developed called the CompTox chemicals dashboard, we see that there are about 900,000 chemicals that are registered in that database. Now, it's important to also clarify here that that database does not only include chemicals that are registered in the United States, includes chemicals that are used across the globe, and it may be also chemicals that are imported into the United States as we import products from other countries. The work that we do at UCSF focuses primarily on pregnancy, and we look at pregnancy because it's this critical period of development where we're most susceptible to biological and biochemical perturbations. I don't know if you've seen this movie before, but I wanted to share this slide. Is one of my favorite movies that talks about chemical pollution in the United States and sort of the aftermath of an environmental disaster in Parkersburg, Virginia. It's a great movie where Mark Ruffalo plays a lawyer that investigates the case of contamination for PFAS in Parkersburg, Virginia, where the DuPont manufacturer plant is based at. One important aspect that comes up in the movie is that chemical manufacturers in the United States are allowed to self-regulate. Now, this movie is set a few years back, and things have changed since then with the new chemicals program, where chemical manufacturers in the United States now, for any new chemicals that they're producing, are required to submit toxicity data and toxicity information to the EPA. But in general, what we could say is that there's a higher degree of self-regulation in the United States compared to other places in the world, let's say the European Union, which has much stricter regulations on chemicals. Another movie I wanted to talk about was more of a documentary. It's called The Devil We Know, and it covers exactly the same story about contamination in the same place in Parkersburg, Virginia. And that documentary covers the story from the aspect of all the tribulations and all the cover-up that went that was happening while we were finding out about PFAS contamination in that area and where people were getting sick. Towards the end of the documentary, there's an interesting idea where they started talking about how epidemiologists that were trying to link PFAS exposures to diseases in the area were trying to find blood samples of people that were not exposed to PFAS. They were trying to find controls. And they looked at several places in the United States. They looked at isolated islands in the South Pacific. It turns out that about 99% of the people on the planet are exposed to these chemicals at low levels, but there's not a single person that is not exposed to them. And finally, what they end up finding is a bank of blood samples that were collected from US soldiers right before the Korean War in the 1950s. And that's about the time when PFOA starts being manufactured at large scale and being distributed. So these were the only samples that did not contain any PFAS. One of the recent stories that has sparked some controversy on Twitter and sparked also a big storm was that the group of the manufacturer of a specific PFAS called Solvay. They decided to discontinue their contract with Wellington Labs for a standard that they were selling. This was particularly important here because these are exactly the standards that scientists use in academia and government to measure these chemicals in the environment, to measure these chemicals in people, in blood samples, and all sorts of biological media. So if we don't have these standards, there's basically no way we can monitor these samples and monitor these chemicals, and no way we can test them for their toxicity. Now, it's been a couple of months since that happened, and finally, Solvay decided to allow the standard to be manufactured by Wellington Labs, but it took a while to get there, and it took several publications and several people talking actively about it on Twitter and other media and calling out Solvay to act responsibly. So I'm going to talk now a little bit about the work that we did at our group at UCSF and our study that was recently published in Environmental Science and Technology. This is a non-target analysis study. It was a pilot study. We're trying to basically develop our methods and try to establish our workflows and pipelines. We started by analyzing 30 maternal blood samples and 30 cord blood samples that were matched, so from the same moms and babies. And we followed, we analyzed these samples with high resolution mass spectrometry. So just to give you an idea of what the workflow looked like, we start with the blood samples and very minimal cleanup because we don't want to lose any important components. We don't do any extraction that would remove any important chemicals from the sample. Since we're doing non-target analysis, we want to make sure that we capture as many chemicals as possible. So once we prepare the samples, and I think for this particular case, we only did precipitation with methanol just to remove the proteins from the sample. Then we analyzed the samples with our instrument, which in this case was liquid chromatography, quadruple time of flight mass spectrometry, which was operated in 2 different electrospray ionization modes in both positive and negative. So the way that works is that you analyze your samples, you get your data from the chromatograms, and then you go through a process called feature extraction and alignment, where you try to see from all that data that you collected, what are the chemical peaks in your samples, and how can you align these peaks across all your samples. So once that process is done, we go through formula assignment. We basically look at the feature, each feature individually, and we assign molecular formulas, meaning how many carbons, how many hydrogens it has, how many fluorines, how many hydrogens, but it doesn't really give you any information on how these atoms are arranged in the molecule. It just tells you what atoms are there. So it's just information on the composition of the molecule, not on the structure. To get more information on the structure, we go through fragmentation experiments, mass spectrometry, MSMS experiments for a selected number of features, where we, in these fragmentation experiments, the molecules break into unique fingerprints, and then we use these fingerprints to match them with databases to get some structural information as to what these compounds might be like. And then finally, for a very small number of chemicals that we're able to confirm, we confirm those with analytical standards. So just to give you an idea of the workflow and the pipeline and how we started working on this analysis. One of the challenges that we are faced usually with non-target analysis data sets is the very large number of chemical features. And it's important to try to find ways to reduce the dimensionality of your data set to a more manageable size. I don't know if you have any good analogies about targeted and non-target analysis. I was thinking the other day that one good analogy is like for target analysis is when you go to the grocery store, you have a list of things that you want to buy and you only buy these things and you get back home and you know what you bought, versus non-target analysis is you go to the store and you buy one of each. And then you get home and you can't figure it out. Like, well, what is this? What is this product that I bought? What does it do? So it's a very similar thing. A similar problem we're faced with when we're dealing with non-target analysis data sets. There's so many features that we have to go through that, in a way, it's almost impossible to go through all of them. So we have to find ways to reduce that data set to a more manageable size. So in this case, what we did was to first of all, look at all of the features that are detected across all samples, so 100% frequency. Then we match that with the peak areas that appear to be relatively high across all samples. And we set a cutoff point at 50%. Oh, sorry, at the 50th percentile. Everything that had a high detection frequency and high intensity was okay to remain in the data set. Then we said that we will only look at features that had some interesting behavior patterns between maternal cord samples. So for example, if the ratio between the maternal cord was higher over 2, or it was very low, or if they correlated very strongly. So anything that showed some interesting patterns between maternal cord was allowed to stay. And then finally, we also wanted to see whether there are any chemical features that show some differences based on demographic variables. So anything that had some interesting patterns with regards to socioeconomic status or education or ethnicity was also allowed to stay. So after doing all that, we looked at the chemicals in the US EPA's Comtox chemistry dashboard, and we excluded chemicals that are already biomonitored by enhanced, chemicals that are present in various drug lists, and chemicals that are likely to be endogenous fatty acids. And that step sort of ensures that we're looking at novel chemicals and not chemicals that have been previously reported. And then finally, we have 2 main steps where we start assigning potential structures to the features that we identified. The first step is the MSMS fragmentation analysis that I explained earlier, which for this particular case, we use online experimental databases, and we also use online fragmentation tools. And for those that we were unable to match with fragmentation spectra, we used an in-house built annotation algorithm that basically assigns molecular structures based on their popularity. And we have evaluated that algorithm with an experimental data set for accuracy in assigning molecular structures, and it has an accuracy of about 75%. So 70%, 75% of the time, the algorithm is most of the time it's right. And finally, we were able to confirm with analytical standards about 17 features, which boiled down to 15 unique compounds. So these are just some figures just to show you what the overall data set looks like. So on the left side, we have the data divided into the negative ionization data and the positive ionization data. And you basically see here the mean peak area ratio between cord and maternal. So you see that there's the 2 ionization modes are fairly similar, but there are some differences. It looks like, for example, that for the negative ionization mode, we have fewer chemicals that are below 0, which in that case means maybe, I guess, that we have fewer chemicals that are likely to partition to maternal blood here. And then when we look at the distributions again between maternal and cord, you see that there's that we have very similar shapes, and overall there's a very good agreement between the levels that we see in maternal and the levels we see in the cord blood. Whoops. This is just to show some results from the demographic analysis that we did. We looked at, for example, race, ethnicity, and this is self-described race and ethnicity, how people identify themselves. And then we looked at maternal education, household income, and nativity, meaning whether they were born in the United States or abroad. One interesting aspect of this figure is that while there is a good agreement between maternal and cord, there are subtle differences when you look at demographic variables, and you can see that certain differences that appear to be significant in maternal samples are not always present in cord samples. For example, you can see here the, differences for race and ethnicity that appear to be significant in maternal samples are not significant in the cord samples. Which sort of gives you an idea that there is there's a reason why we see these differences, and probably that reason is how chemicals partition between maternal and cord blood, how chemicals behave with regards to their ability to cross that barrier from maternal to cord blood, which is more likely to be the placenta. And there are several studies, both pharmacological and environmental chemistry studies, that have reported on that function of the placenta to regulate that partitioning of chemicals. So we're going to get back to that, but just to take a break here, and I'm going to talk a little bit more about non-target analysis. For our annotation scheme, we used the proposed framework by Szymanski et al., which basically groups annotations based on their confidence. So starting from the lowest level of annotation, level 5, that's when you only have a mass, an exact mass of interest that you have measured with your instrument. Level 4 is when you have an unequivocally assigned molecular formula. That means that for that feature, there are no other formulas that could be matching. You have one single formula. Level 3 refers to tentative candidates, and usually in level 3 we have MSMS matching with in silico databases or any other diagnostic evidence through annotations, through annotation algorithms or through literature that you have collected that can help you to have some diagnostic evidence as to what that chemical might be like, but there's not one single candidate that that would be the best candidate. You have multiple candidates for the same feature. And then finally, level 1 and level 2. So level 2 is probable structure based on a spectrum match with experimental MSMS databases. And finally, level 1, a confirmed structure with the highest analytical standard through matching with analytical standards. So this is a couple of figures here to show you the chemicals that we found in our samples. On the left side, you see that we have a bar plot for all the compounds that we detected, and they're grouped by levels of annotation, level 1, level 2, and level 3. You can see that we have chemicals across all spectrums, all across the spectrum of uses and applications. We have pharmaceuticals, we have pesticides, flame retardants, PFAS, plasticizers, cosmetics, chemicals used in consumer products, chemicals that are high production volume chemicals. And what's very interesting also here is that we have a large group of chemicals that could not be grouped in any category because there's very little to no information about their uses, and they were grouped as unknown. Now, on the right side of the figure, you'll see what happens when we remove all the chemicals from that plot that were previously reported. So we're left with a few pesticides, a small number of PFAS, some plasticizers, and a very large number of unknown chemicals that have very little information on what their applications are. So this is particularly interesting because it sort of points to the fact that not only we're unsure about how many chemicals we're being exposed to, how many chemicals are out there, but even when you find these chemicals, you're not always 100% sure as to what these chemicals are being used to and how exactly you're being exposed to them. So a couple of examples of what these chemicals look like. Some of the chemicals that we found. Is, for example, the one chemical on the top right, which is a chemical that most likely is used in consumer products and specifically personal care products and fragrances. And on the right side, we have a plasticizer that is used in the production of plastic polymers. Now, these are very, very interesting chemicals because they're not regularly monitored, and there are not that many studies reporting on these chemicals. And because they're so ubiquitous through their applications, we can be fairly confident that they're also spread out across the population. A lot of people are exposed to them. A couple of other interesting chemicals were these two PFAS chemicals that we found. The one on the left is likely an impurity of PFNA, and it's most likely used in fluoropolymer manufacture. It could also be a breakdown product of long chain fluorotelomere based products. And the one on the right here is also another PFAS, most likely coming from products used as surface protectors in textiles, food contact materials, carpets, and etc. These are two of the chemicals that we have very little information about as to why, what they are and how exactly they're being used. I mean, we can make educated guesses based on their structure and based on their similarities to similar chemicals, but it's interesting that we don't really have a clear picture. And also, part of the reason that may be is that maybe they're not the main active ingredient. For example, is for the left, if they're the impurity that comes from the manufacture of the main ingredient and they're carried over in the product, it sort of makes logical sense that we wouldn't have a lot of information about them because they're not the main ingredient. So whoops, yes, there we go. Then I wanted to present some preliminary data from one other study that we did that followed up from this non-target analysis study. And one interesting aspect of these non-target analysis studies is that not only they can tell us about what chemicals we're being exposed to, but they can also inform other in silico and in vitro studies as to what chemicals should we look for. For example, when we look at the partitioning between maternal and cord that we talked about earlier, what chemicals should we focus on based on their behavior to partition, likely to the cord blood or, not being filtered out by the placenta and therefore pose a risk to the fetus. So for this case study, we looked at PFAS that are registered under EPA's chemistry dashboard, and there were about 9,000 chemicals that were registered in that list. That list it contains chemicals that many people know, like the PFOA, the one on the left. It also contains novel PFAS, like the one in the middle, Gen X, and it also contains PFAS that very few people have heard of, like this perfluorocyclohexane. So a few words about how we went about this process. We decided that we wanted to build a model that would be able to help us make predictions for the tendency of a chemical to accumulate more towards the fetus or to not being filtered out by the placenta. So for that purpose, we collected data from the literature and we built our database. We collected measurements of the ratio of the maternal cord from the literature, from matched studies that measured both the concentration in maternal samples and the concentration in the cord samples. We prioritized the values that we collected starting from geometric mean. If that was not available, then we went for median. If that was not available, we went for the arithmetic mean. And because there was a lot of variability on how people report these values as sometimes lipid adjusted, sometimes non-lipid adjusted, we collected everything and then we back calculated from the lipid adjusted measurements the non-adjusted, and we completed our data column with non-adjusted measurements of the partition ratio in maternal and cord. So finally, we collected the chemical identifiers for these chemicals, and from there we collected their linear representations, smiles, and their physical chemical descriptors. For this exercise, we used Mordred, which is a package that can be used to calculate over about 2,000 descriptors. And then we tested 3 different architectures. We tested 3 different machine learning models for their ability to predict the partition between maternal and the cord, and we evaluated these models. And then finally we used the validated models to make predictions for the PFAS that are registered under EPA's chemistry dashboard for the 9,000 PFAS. So these figures here show the results for the three models. The left is an artificial neural net. The middle one is a random forest, and the right one is a support vector machine. You see that there are similarities in how these two models perform. They all estimate that the majority of PFAS will be around 0 log RCM, which means basically will be around 1-to-1 ratio, 1-to-1 concentration between maternal and cord. So if you measure, for example, 50 nanograms per liter in the maternal blood, more likely you'll also have 50 nanograms per liter on the cord blood. You can see that for a very large number of PFAS, especially in the case of the neural net and the random forest, there will be PFAS that will partition preferentially to the cord blood. About half of the PFAS in the database are preferentially partitioned to the cord blood. And to put that into perspective as to what that means, I used also two pharmaceuticals as benchmarks that are considered fairly safe and are commonly prescribed during pregnancy. These are SSRIs, antidepressants, that are considered to have very little effects during pregnancy, and they're considered fairly safe. Part of the reason why they're considered safe is that their partitioning behavior makes that they partition to the maternal blood over 3 orders of magnitude, 2 or 3 orders of magnitude higher concentrations compared to the cord blood. So, and what's interesting also about these pharmaceuticals is that they're actually fluorinated. So if you see here, they both have a carbon that is fully fluorinated in the top of the benzene ring. They wouldn't be classified as PFAS, at least according to EPA's definition of PFAS, because they only have one fully fluorinated carbon. But under other definitions, they could be. And if we have any pharmaceutical chemists in the audience, you understand why these fluorines are there, because they increase the affinity of the chemical to the target protein. And if we have any environmental chemists in the audience, you probably see why this is a problem, and because these bonds are so strong and they're impossible to break. And these, even though they're pharmaceuticals and we benefit from them, they could potentially pose a risk to the environment. But yeah, this is just mainly just to show you. I just wanted to use these two compounds as benchmarks to sort of illustrate where we are with PFAS with regards to known relatively safe pharmaceuticals that are used during pregnancy. And as you can see, in all three cases, we're very far off from what is considered to be fairly safe. Over 3 orders of magnitude difference for PFAS. There are no PFAS that are in that safe region of these two SSRIs, these two antidepressants. Finally, we looked at what are the functional groups of these chemicals, what are their properties, their structural properties that define their behavior. So we'll look at functional groups of chemicals in our collected training database that showed some positive association with RCM, meaning that they're likely to partition more to the cord blood, or chemicals that showed a negative association with RCM, meaning they're likely to partition more to the maternal blood. And what you can see here with yellow, we show aromatic carbons, and with dashed lines, we show the bond between two aromatic carbons, so basically the approximate electron distribution between these two carbons. You can see, for example, in the chemicals, the top panel that are expected to partition mainly to the maternal blood, we have a lot of polar groups, functional groups that make the chemicals partition, that make the chemicals be more hydrophilic. And there's an interesting idea behind this, and that is that there's a difference between the lipid content of the maternal and cord blood. The maternal blood has about 3 times more lipids than the cord blood. So if a chemical likes to be more in water, it's more likely to cross the placenta and partition preferentially to the cord blood. versus when you look at the chemicals in the bottom, these are highly hydrophobic groups. And it makes very good sense that they would prefer to partition more to the maternal blood because it has more fat than more lipids than the cord blood. So that was for the collected database. And now when we look at PFAS, we see an interesting story here. We see a very similar pattern for the predictions that we made. So for the chemicals, for the PFAS that are expected to partition more to the cord blood, we see the high presence of polar groups, such as alcoholic groups, carboxylic groups, and ether groups connected to fluorines, versus for the chemicals that are expected to partition primarily to the maternal blood. In the bottom, you see more aromatic bonds connected to fluorines. We have the triple bonded nitrogen, which is a polar group, and it's a little bit of an outlier. And the sulfonic group here in the end, but it's interesting that we see similar behavior between the two databases. A lot more polar groups on the chemicals that are partitioning preferentially to the cord blood, and fewer polar groups in the chemicals that are partitioning preferentially to the maternal blood. Yeah, and I think that's all I had to say for today. I want to thank you all for coming to this presentation, and thank our sponsors for these projects. And if you want to hear more about us, you can follow us on Twitter at Dimitri Abrahamsson or Tracy Woodruff or UCSF Pre or Earth Center. Thank you.
Thanks, Dimitri. That was fabulous. We have a question in the chat about the results from the non-targeted analysis. Do you see the chat?
Oh, okay. Yes, I just saw it. Yes. Were there any compounds that were exclusively in maternal versus cord blood? Yes, I think they were, or at least apparent to be exclusively in maternal versus cord blood. And then the reason why I'm saying apparent is because the instrument that we're using, QTOF, has a high resolution and in terms of mass accuracy, but it lacks in sensitivity in terms of detection limits. So they may appear to be present in one matrix or the other, but it could very likely be that it's just an undetectable amount. Oh, there's another question. Why were only fatty acids screened out? I don't think that there weren't. We filtered out some fatty acids, but there were other chemicals that were screened out too, like chemicals that have been previously reported and regularly monitored by Haynes. The reason why we took these filtering steps was to ensure that when we downsize our data set, we're focusing primarily on chemicals that are novel and have not been well studied, have not been well monitored in the population.
You should talk about what you found in the larger data set with the PFAS and the fatty acids.
Right. So in a follow-up study where we examined the associations of exogenous and endogenous chemicals, basically we wanted to see whether there's anything we can learn by looking at the interplay between the exposome and the metabolome. So whether there's any interplay between exogenous chemicals and endogenous chemicals. We found some interesting correlations between PFAS and fatty acids, which could be indicating what other epidemiological studies have found, some lipid dysregulation metabolism to people that are exposed to higher concentrations of PFAS. It could also hint to some molecular mechanisms that could involve fatty acid binding proteins and PFAS. I think this goes back to what we were saying about non-target analysis, that these studies are great because not only they can inform us about what chemicals we're being exposed to, but they can also generate hypotheses that need to be tested in the lab, in vitro, or they can generate hypotheses for biomonitoring studies.
Yeah, I want to also follow up because Susan asking about the endogenous lipid classes, and I think we've talked about this, right, Dimitri? But the molecular size is not optimal to capture
Right.
some of those of the that we're scanning for. Is not optimal to capture some of those, all of them, I guess.
Right. That's also an important point, that where our analysis primarily focused on low molecular weight on small molecules. So if we want to do a more comprehensive analysis on the lipid side, we would have to basically shift the mass range window that we use to look for chemicals in our analysis, which would be interesting and definitely worth looking into. Just we're primarily focusing on small targets, small molecules.
So there's some questions from Jennifer about, I hear there are so many unknowns, Dimitri.
That's interesting, Tracy. I wonder why there's so many unknowns. Because I guess
I'm sure EPA will reveal that soon. Anyway,
right, because that goes back to the idea of self-regulation, because there's a much higher degree of self-regulation in the United States. And when that happens, there aren't any requirements to report uses or applications for chemicals you're producing. So I would say that that's the main reason, that there is no requirement to disclose that information. And there is for some chemicals, there is enough information, but for many others, it's a fishing expedition. We have to figure it out.
Right, it does vary by law, right? Like, so pesticide
It
usage
varies by law, exactly, yeah.
has much more information requirements. Jennifer also asked, were there some compounds and concentrations that seem unusually high? And before you answer that, just to go back to the maternal cord partitioning, you also don't, like, this method doesn't capture certain types of chemicals or pollutants, so metals, for
Right.
example, and we know like methyl mercury can bioaccumulate, and cadmium doesn't seem to cross the placenta at all. So there's also other groups outside of this that are not
Exactly,
captured
right.
anyway. So
We would need a different instrument, a different type of analysis to capture metals like ICP-MS, inductively coupled mass spectrometry, plasma mass spectrometry. So that would also be very interesting, since we were looking at the whole exposome. And I know that there's been a couple of studies that looked at both organic chemicals and metals, but it's also a whole different story.
Right. Did you find anything very high things, very high levels, very high concentrations?
Very high concentrations. Well, we haven't been able to quantify everything. So, these non-target analysis studies are primarily non-quantitative. We were able to quantify a small number of chemicals, and some interesting findings that we saw were high levels of tetraethylene glycol, which is a chemical that's been used in all sorts of applications because it's a very useful surfactant. It's used in consumer products and detergents and things like that. And this is something, to my surprise, when I was looking at that chemical, I couldn't find a lot of toxicological studies, which is quite interesting because it's so prevalent. You'd think that there would be a lot of information out there. It doesn't look like it.
Do you see there's did you want to, could you talk a little bit more about the numbers on the, that you had those numbers on the functional groups? Basically,
right.
That you had functional groups that partitioned more in maternal and more in the cord blood on the last two slides.
Maybe I can bring up the slide. Oops. Yeah, these slides, right?
These the numbers here, 573,929, and the percentages.
Right. So the numbers are basically, we use a computational chemistry tool called RD-Kit, which basically assigns specific numbers to specific functional groups. So, and essentially every functional group has a unique identifier. So the top number is the identifier for that group. The percentages, though, are interesting because they are the frequency of that fragment in the database. So it gives you an idea as to how frequent that fragment
in the RD-Kit database.
In our collective database.
How often this functional group shows up
in our data
chemicals we're analyzing?
Exactly. Yes.
Okay. So this one over here on the right shows up 44%, like almost half.
44% have an aromatic carbon somewhere in the molecule, which kind of makes sense because it's a very fairly common group.
Right.
And then the more, the more you go towards very specific groups that are fairly rare across molecules, these percentages drop.
So what's an example of a rare functional group?
So, for example, the one right here, which is the one in the bottom right, 74.
Oh, that's a 3%.
Yeah, that's a 3% because it has an aromatic carbon, two aromatic carbons connected to each other, and then it has a sulfur connected to it. So, for example, that's a fairly uncommon functional group. You can, for even the chlorines are not that common compared to the simple aromatic carbons. So chlorines connected to aromatic bonds are not that common, at least in our database.
Someone's asking about air pollutant chemicals. Do we know if they cross placenta?
Air polluting chemicals like PAHs, like polycyclic aromatic hydrocarbons, these kind of chemicals. I'm not sure. I think we have some chemicals like that in our database. From what I remember, because they're very hydrophobic, they would preferentially partition to the maternal blood. But I'm not really sure. That might be something I need to double check. It's just an educated guess based on the structures, unless we're talking about other air pollutants like particles.
Does look like watching real-time machine learning happening in your head.
Yeah, there's a lot going on there.
Okay, so I wanted to say something else too about this, and I can't remember what it was because my dog barked. Yeah, I don't know if anyone else had any other questions. I will say that in part, not the only people, but there has been a lot of activity to ask EPA or request that EPA require analytic standards as part of the registration of new chemicals so that we have more information, which they do actually under the FTPA, which is the law that regulates pesticides, pesticide active ingredients. So I think we're going to see more movement in that area because EPA seems to be a little bit more responsive to this idea at the moment. So I think that's a really excellent example of something that may seem a little bit obscure, analytic standards, though I know not to analytic chemists like Dimitri, but has a really important public health regulatory policy implication because it is one of the things, like we see Jennifer's on here, and Josh, that we've been doing is taking some of these chemicals and looking at testing them in vitro systems to identify different types of developmental effects like reproductive health effects or neurotoxic effects. And sometimes you cannot even purchase these chemicals because sometimes manufacturers are not allowing them to be sold, but also they may not even be available for purchase in the United States, even though we may be exposed to them. So hopefully this will be an opportunity for EPA to improve that element of their policy program. So I want to thank you again, Dimitri, for speaking.
Thank you, Tracy.
Super interesting. We work together. I learn something every time you speak. So
me too.